Adam Jones. I'm a professor at the University of Idaho. I was at Texas A&M University for a while, so over a decade now at University of Idaho. Yay. Go Vandals. So today I'm going to talk about some work that I've been doing with Steve Arnold and Reinhard Berger over about the last, sort of evolved over about the last 2 decades or so. So it's kind of a long project that's been going on and on. And the issue that I'm going to address today, though, is how do things like pleitropy and epistasis affect local adaptation and our ability to detect local adaptation in genome-wide scans, population genomic scans of variation across the genome. And let's see what my next slide is. All right. So before I get into that, so imagine that the situation is that we have 2 populations. They occupy different geographic locations. There's divergent selection in those 2 locations, and it's acting on polygenic traits. Right? So under those circumstances, you might get local adaptation. Generally, when we deal with these polygenic traits, we use strictly additive models. And so you might imagine that other genetic architectures that deviate from that additive model could result in different abilities to detect these signatures of local adaptation. Does that make sense? Okay. And so the question was, if we start to get into pleitropy and epistasis, is that going to affect these genome-wide scans of local adaptation? I'm going to focus mainly on epistasis in this talk. And the reason is that epistasis is the more neglected topic out of the 2 between pleitropy and epistasis. The reason for that is that pleitropy is pretty easy to deal with. Epistasis is more difficult. There are 2 types of epistasis that you kind of need to know about. So one is functional epistasis. So this is going to describe the actual molecular interactions between genes. And that's going to be the same regardless of the allele frequencies and things like that in populations. Think of those as genetic pathways. On the other hand, we have things like, we have statistical epistasis, which is going to describe any kind of non-linear or non-additive interaction between loci. And generally that's measured by looking at breeding designs and estimating the non-additive components of genetic variants. Okay, so just because there's functional epistasis doesn't mean there's a whole lot of statistical epistasis. And that's something to keep in mind because statistical epistasis depends on variation in individual loci in the population and allele frequencies and things like that. But what we know, and that's definitely true and not really up for debate, is that functional epistasis is basically everywhere, right? And so we know this from developmental biology, from molecular biology, from evolutionary biology, regardless of the field. The fact that the genetic background matters when you do experiments involving mutant individuals means that functional epistasis is everywhere. As people start looking more into gene networks, you have a view like this highly interconnected series of gene networks, and then the output of that is some kind of phenotype, right? So they're really complicated. There's a lot of interactions. Even labs have epistasis in the coloration. All right, so if we go, okay, epistasis is everywhere. How important is it? All right, and so what I'm going to address in this talk is just this question of how does it affect detecting local adaptation. There are lots of other effects of epistasis, and I'm not really going to go into here because it would take like hours and hours to go into everything that epistasis matters for. And so I'm just going to focus on this one problem. And so what you can imagine is that if there's lots of epistasis versus no epistasis, just strict additivity, you might get different patterns of divergence of those polygenic traits when they occupy different environments. Or another possibility that seems likely is that if you think about epistasis, in addition to having direct effects on the trait, there are going to be all these gene interaction effects on trait values. That makes it seem hopeless to ever detect loci that are involved in adaptation because the adaptation is going to depend not just on the direct effects of the loci, but also their interactions with all the other loci. And if you think about pairwise interactions, just pairwise interactions, and you have 50 loci, there are thousands of potential pairwise interactions. And then there are higher order interactions as well, which makes it even worse. And so it's sort of, when you start thinking about epistasis, just doing thought experiments, it seems like a hopeless problem, right? And that was part of the motivation for this study. All right, so what we did is just a pretty simple case where we just did individual-based simulations of two populations linked by migration, and they have different optima for some polygenic traits, right? So it's a pretty simple demographic scenario. The model is just an individual-based simulations, so it has a life cycle. You start with adults, they mate and produce progeny. We keep track of individual loci. We keep track of individual alleles of those loci. There's Mendelian assortment. There's some number of progeny that are produced. There's selection according to a Gaussian individual selection surface. Some individuals survive that selection, and then there's some population regulation, which is just a hard carrying capacity. So individuals are randomly culled until you have the carrying capacity. We've used these models in the past to study the dynamics of the G matrix, and those are strictly additive models, but there's a number of papers you can look at to see what the sort of details of the models are. We explicitly simulate all individuals and all loci, like I said. And then there's a whole bunch of parameters, like the number of quantitative trait loci, mutation rates and mutational distributions, the selection surface, the population size, environmental variance, so on and so forth. Everything you can imagine in a normal population. So the big extension here from the additive model is to model epistasis. So we just use the multivariate model, and basically that's pretty easy to implement. So in addition to direct effects, additive effects of the alleles, you have interactive effects, right? So it's looks kind of like an analysis of variance. And then each interaction will have an epistatic coefficient. There will be lots of those epistatic coefficients because there are lots of gene interactions, so we just draw those from a normal distribution. All right, like I said, it's two populations connected by migration. We have any combination of pleiotropic, non-pleiotropic, and epistatic loci affecting the traits. We have any number of marker loci, and we explicitly model the chromosomes, including recombination events, with the marker loci on those chromosomes. Okay, so the first result, so what I'm going to do, instead of showing a bunch of tables and stuff like that, I'm just going to show some representative results in figures, and then kind of extrapolate to the general results. So the first thing is you could say, okay, well, how much does epistasis affect local adaptation? So now we have two individual selection surfaces shown here. That's the orange. And then the G matrix is basically drawn on here. So the distribution of genetic variation in breeding values is shown as an ellipse. And space here represents phenotypic space. So that's the distance in the optimum between the two populations. Population 1 is over here, and population 2 is over here. And so what you can see is that with one migrant per generation, each population is displaced from its optimum a little bit toward the other population from which migrants are coming. Does that make sense? All right, so they are locally adapted, and migration moves them away from their optimum a little bit. And then what happens if we increase the variation, or the migration rate? And so what we see is that as there's more migration, they're displaced further from their optimum, which totally makes sense. And this makes sense in an additive model. The other thing we see is an increase in the genetic variation along that axis of selection. So the top is without epistasis, and the bottom is with epistasis. There is a difference. It's sort of subtle. There are two differences. So the first difference is that with epistasis, they're not displaced quite as far from their optimum by migration. So it's a little bit hard to see, but the ones with epistasis are a little bit closer to their optimum, right? That's not a very impressive effect. The other thing that's happening is without epistasis, you can see that there's an increase in additive genetic variance for trait 1, the one that's on the axis. There's another trait, there's an x-axis trait and a y-axis trait, is trait 2. So the G matrix is stretching along that trait 1 axis, but it's not getting thicker without epistasis, right? So if you look here, it stays the same width. And that's because trait 2 is not affected. The variation in trait 2 is not affected by this migration. Whereas with epistasis, it is. So you can see that in general, under epistasis, the entire distribution of genetic variation for both traits is increasing. So you're increasing variance in trait 1 and trait 2 through migration, even though trait 2 isn't under selection. That happens because trait 1 loci with epistasis now epistatically affect trait 2. And so anything that increases variation in trait 1 also increases variation in trait 2 through those epistatic interactions. All right, so those are two effects. But now let's get into sort of the main issue that we wanted to deal with, which is, can you detect loci through genome-wide population genomic scans when there's epistasis in the genetic architecture? Okay, so I'm going to just show some of these Manhattan plot type things. So we have marker position across the genome. We're just saying there are 2,000 markers in the genome. This is pretty general. We could increase the number of markers and get kind of the same effect. We have FST on the y-axis, and then the QTLs are shown as the colored dots, and the gray is the marker loci. So if there's hardly any migration, even if there's strict additivity, you don't detect outlier loci, and it's because there's so much divergence due to drift that they're just buried in the distribution of marker loci. So the QTLs don't diverge enough that you can detect them in the background. But if we increase the migration rate, so that it's an exceptionally high migration rate, then those outlier loci start to appear. And so what you'll see here is there are a couple of QTLs that are strongly diverged, and in those regions of the genome, there are marker loci that are also strongly diverged. And so those would be things that you would detect in a genome-wide scan for selection. What about when we end epistasis? Well, you know, it's kind of the same. And so, okay, and so it's kind of the same, except now sometimes the outlier loci are actually loci affecting the other trait. So it's a trait 2 locus that's different because it indirectly affects trait 1. But it's kind of the same. With pleitropy, everything's basically the same as additivity. So I'm not even going to waste any time on that right now, especially since I'm out of time. And then if we look at the just the difference in the FST values overall, we see that there's a higher FST without epistasis than with epistasis, but it's kind of a small difference. And so the conclusions are that first, there could be lots of epistasis even if you have a lot of additive genetic variants. This is just a general point about epistasis. Regardless of the genetic architecture, trait divergence almost always involves just a couple of loci. So even if we have 20 QTLs, there will be like 2 that are strongly divergent. Those loci are generally detectable using genome-wide scans for QTL, but only when selection is very strong and migration is very high. And epistasis does have a quantitative effect. It makes it a little bit harder to detect, but it's still possible to detect those outlier loci, even in the presence of epistasis. The signal's a little bit weaker because it's distributed across more loci, but for the strongly divergent loci, which also occur in epistasis, those signatures of selection are detectable. And so it's kind of good news, right? So epistasis doesn't make genome-wide scans impossible. If you want to read this, we already have a paper that's in press in Journal of Heredity, and so all the details of the model and more results are in that. And you can download the preprint from the Journal of Heredity website. The title's basically the same as the title of my talk. Okay, thanks for your attention. That's it.